Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 150th seminar, and I call it a Rice University Day. Happy New Year, and I cannot believe more than 2 weeks passed this year already, and I hope you will continue to keep your New Year's resolutions. Since the SynBYSS conference in Hawaii, I haven't traveled at all for 1 month, although I had a great time in San Diego with my family and also enjoy my seminar visit to UC San Diego yesterday. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jonathan or Joff Silberg at Rice University. He is the Stewart Memorial Professor of Biosciences and also the Director of Systems, Synthetic, and Physical Biology program at Rice. He is a pioneer in bioelectronics and environmental synthetic biology, as you may know, leading multiple cool projects, and some of which also led to big journal publication recently. I have uh, known him for many years, and it's great to have him today as the first speaker of 2025. Uh, Geoff, thank you so much for your contribution to scientific advances and education, and also for starting or kicking off this, great 2025. And the virtual podium is all yours now. And thank you again.
Awesome. Well, while I share screen, I do want to thank you for inviting me to come here today and speak with Reza. And thank you for organizing this online, virtual, and in-person seminar series for the community. I really appreciate this. I know many folks that I've talked to really appreciate the program you put together and how you help weave the community together. And so in my short talk today, I really want to talk about where we've started to shift in our thinking for synthetic biology. And I think Taesuk also works in this space, and I really appreciate what he's been doing. But we've been thinking a lot about developing tools for top-down programming of undomesticated microbial communities. And I think the reason we've really thought about this is we've been thinking about how to accelerate the design, build, test, learn cycle for applications that support the bioeconomy. And I think there are two major challenges. One is that top-down genetic programming of environmental communities is just hard still. And the second is if you start programming communities, it's hard to control protein expression and achieve it reliably in specific microbes. And so I want to talk about some of the tools that we've been developing. And I think the tool I'm going to talk about today is really aimed at addressing this question, which is we don't understand the host range of mobile DNA, which really limits the direct programming of microbial communities. And it's not that we don't understand some of the host range, but we really don't have algorithms that predict where a coli-1 origin is going to function when we modify plasmids. And so we're very interested in this for many environmental communities, soil in particular. But what I'm going to talk about today is a lot of work in wastewater sludge communities that we've gotten from a local wastewater treatment plant near Rice University. And this project I'm going to talk about is highly collaborative. This is a project that I've been doing kind of equal contributions from my lab, Dr. James Chappell's lab, and Dr. Lauren Stadler's lab, and many graduate students. And our idea was, could you store information about mobile DNA host range using ribozymes? And could you store information across a community, not just in one microbe? And as we were thinking about this, we really started to think about what are the conserved biomolecules where you might want to write information. And we kind of started with something we thought would be very high risk, which was 16S ribosomal RNA. The reason we chose this is because environmental scientists sequence the variable regions to say who is in a community. So what we wanted to do is to sequence who's in a community and then write information with a catalytic RNA that we've engineered that tells you who participated in gene transfer. And so you can think of looking at the 16S ribosomal RNA as a passport that marks where the mobile DNA is traveling. And then we're going to code the catalytic RNA as the stamp that puts information in that 16S ribosomal RNA. We thought this would be very risky because this is a structural RNA, stable, has armor of proteins, and we were really shocked that it worked. And so what I want to tell you is just briefly how the barcoder works. We use a very simple genetic circuit, much simpler than much of the work we do in the lab. Single promoter making a single transcript. The transcript encodes a catalytic RNA built on the tetrahymena ribozyme. It's got a core that's the catalytic bit. It's got an RNA guide that's much like a CRISPR guide. This says where is it going to anneal using Watson-Crick base pairing, and then a barcode, which is the information that's going to be stored. When this is transcribed in a cell, we create guides that direct it to ribosomal RNA. It will then anneal. And if there is a uracil where we programmed it to anneal, you will get catalysis and barcoding. And you could do this with 16S ribosomal RNA. You can do it with other types of RNA, but this is kind of where we started with. It ends up that this works pretty well. I'm not going to show a lot of work we did to just prove it works in E. coli, move it into different microbes. But once we showed that this works in gram-negative bacteria, we went and created an E. coli with an RP4 mating pair formation system where we coated our ribozyme into a small plasmid that this would conjugate. We acquired sludge from the West Houston wastewater treatment facility and mixed them together. And, it's not in situ, it's bringing it in the lab onto agar plates and just incubated them for a day. And then we sequenced using kind of cheap, short read sequencing who's there and who was barcoded. And after we developed a computational pipeline to analyze this data, we found that for this shallow, about 1 million read sequencing, we found 279 amplicon sequence variants, and 50% of them presented a barcode. And so that was very exciting when we got this first data, suggesting that this can store information across a community. We've done a lot of work that I'm not going to show validating this, comparing this against fluorescent-based reporters, and the signals mirror what you get from traditional kind of approaches. But what we've really done recently is to diversify it. So we've made multiple plasmids with different origins and different barcodes. So now we've got stamps for both of these different plasmids. We put them into the same RP4 mating strain, mix those together initially, and ask the question, when you sequence these mixtures, is the barcode, barcode 1 or barcode 2, proportional to what we mixed? And in fact, you see that. So in a single sequencing run, we can now start to look at multiple mobile DNAs. Now, this is just a mixture. It's a simple mixture of the same E. coli, just transformed with different vectors. How about in a community? So what we did after that is we went into a community and we took 2 different E. coli. So the E. coli are identical, except they've been transformed with different conjugated vectors. The vectors differ in 2 things, their barcode and their origin of replication, and nothing else. So you mix E. coli with wastewater sludge again. This is about a year later. We got almost the same number of amplicon sequence variants, within about 10% from the same wastewater treatment plant. We sequenced who's there and who has barcode 1 and 2. And this is really exciting because what we found is about half of the organisms had both barcodes, and some were more specific to PBBR1 versus COLI1. One thing that was interesting is the total fraction of the microbes we were able to barcode went up when we watched 2 mobile DNA in the same time. So you could imagine, like, add another, you could start pushing up the fraction of the community that you start hitting in a single experiment. You could also start learning about host range and kind of, mapping that by doing these experiments again and again. Oh. So really what I wanted, my take-home message is we build very complex circuits. So we actually have built like 8 component synthetic electron transport chains. And those are fun and really are cool for kind of bioelectronics applications. But I think it's very interesting to think about organism space and maybe making your circuits very simple and starting to learn how do you program across more diverse organisms in a single experiment. And I think the tool we've built, which we'll soon be sharing on AdGene because the paper was just accepted, that describes this, will be very powerful for any type of gene transfer you're interested in: conjugation, transformation, transduction. And in fact, we started to do some work putting this into other types of mobile DNA. And I do want to emphasize, maybe for the broader community, that this work is really required a team. You know, it's not just my lab. This has been a true collaboration between James Chappell, Lauren Stadler, and my labs. Lauren is a wastewater microbiologist. James is an RNA synthetic biologist. I'm an environmental synthetic biologist. We could not have done this without the teamwork. And maybe that's for the more junior folks. I think it's been really awesome to work in teams, but I think that's really allowed us to push on this kind of complex community programming. And of course, thank you to the agencies that supported us, as well as to you all for your attention, and Tae Suk again for the invitation.
This is fantastic. Thank you. I now see multiple more impact out of this work. You just mentioned this gonna be good tools. At the same time, what I'm thinking from my perspective, because I'm kind of also a little bit worried about, of course, or a lot worried about the conjugation or horizontal gene transfer that kind of spread the antibiotic resistance, and also the worry about we dumping too many synthetic DNA into the sewer or from the lab, and then this probably elucidate mechanism or speed of the DNA transfer in the real environment. This is a fascinating work.
And super spreaders. I mean, I think it's really interesting to start thinking about the networks and,
Yes.
could you start understanding who
Yeah.
maybe amplifies thing or potentiates it? Yeah.
Yeah. Yeah. So that is a fascinating one. I potentially want to collaborate with you about. I'm thinking about huge project, about this kind of distribution of this, element, and then transfer to another one in horizontal by horizontal transfer. This is a fascinating work, and then this is exciting. Thank you so much.
Thank you.
Yeah. All right. So fantastic. Okay. So now the main speaker of today with a longer introduction, Professor Reza Zadegan received his bachelor degree from Karachi University and also his master from SynBYSS University and his PhD in nanoscience from Aarhus University in Denmark. After coming to the US, he has been postdoc at the Markey Cancer Center of the University of Kentucky and Micron School of Materials Science and Engineering of both states. He is currently an assistant professor in a tenure track position in the nanoengineering department of the Joint School of Nanoscience and Nanotechnology and at North Carolina A&T State University. NC A&T State University, which is the largest historically black college and university in the nation, the department and the joint school actually hired him to help create technical and educational bridge between the contributing disciplines and synthetic biology. His long-term goals have guided his research direction, which integrates nanotechnology, information technology, and synthetic biology. In collaboration with industrial and academic leaders, he has co-authored the semi-symbio roadmap that drives research and development at the intersection of synthetic biology and semiconductors. The semi-symbio roadmap and the identified NSF board ideas, I believe I also saw that one, several years ago, that have directed his career path and educational and research trajectory. His expertise in synthetic biology, computer sciences, molecular biology, molecular modeling, DNA, nanotechnology, microfluidics, and DNA editing enables him to pursue and lead the proposed activities. His goal is to position his university among the leaders of this emerging field, and also as such, he is actively engaged with industry, academic, and government stakeholders, and he designs, develops, builds, and tests concepts related to DNA-based memory, computation, and nanotechnology. Finally, by incorporating research into courses, he has created a pipeline for students to engage in hands-on, inquiry-based learning experience, preparing them for careers in STEMs. Recently, I also recruited him based on his excellence to my NSF Global Center, starting to work together to realize his vision. Reza, thanks so much for your time today, and please take it away. It's all yours now. Thank you.
Thank you, Tarsek. It's a great pleasure to be here. I second Joe on uh, thanking you for this great opportunity and also for being the pioneer for starting the seminar series and um, yeah. So it's very hard to talk after Joe, of course, because you see very great work, and I try to now show that I have something to say as well. So hopefully, hopefully there will be something interesting for the audience. Now, aligned with what Joe said about programming materials and biological materials, my lab also is focused on doing the trying to program materials, but mostly we work with DNA. So some of the applications of DNA for synthetic biology, I'm hoping that today I can present some of that and would be of interest to the community. Now, Geoff was talking about a kind of memory that you would want to have for cells so that there will be communication between the organisms. But today, I'm also showing another side of the synthetic biology and the applications of a molecule like DNA that is a carrier of information. Aligned with this semi-synbio, the original thought was that we might be able to harness biological materials for electronics as examples that Joe mentioned. And there is another example that I'm going to present today, and that would be on storing information on DNA. And I'm referring to digital information and also some other applications of DNA that we may be able to use for biomedical and biomedicine. With that, so to give you an idea about why the programming materials is important, I would like you to look at this slide where it shows basically two types of mobile phones. On the left side of the screen, you would see a couple of strong men carrying two mobile phones or phones that now we can call mobile phones because they are carrying it. That's an image that we have from 100 around 150 years ago. And on the right side, you see my son holding a phone and trying to connect with me. Now, the difference between these two devices is that the device on the right side is very small, at the same time has a lot of capacity for doing computation, taking images, do a lot of things, including storing information and communicating between or providing the communication between people. On the left side, though, those two phones only connect two houses that at the moment were probably only a few hundred yards from each other. Whereas on the right side, that phone alone connects people from uh, countries, continents, and cities, and so on. So all of that has been only possible because the semiconductor industry was thinking about how we can reduce the size and increase the capacity, and that's the promise of Moore's law, where you would expect that happen. But we're at the end of Moore law, and that's where the community that is driving the semi-symbio, came up and thought that maybe we can recruit other molecules other than the normal silicon-based materials for biology. So majority of the work that we do in my lab is focused on dealing with big data and managing that. And at the same time, the second part of the
Reza, your volume has gone down a little bit.
Yes, I want to say that one. Can you close a little bit to the computer?
Yeah. Is it better now?
Yeah, better now. Yeah.
Okay. Yeah, try to stay close to my microphone. Yeah, so the second part of the research, or the second category of the research that we do, is based on trying to develop the technologies that enable smart and intelligent medicine, where you would be able to just target specific diseases and try to deal with them. And there are other applications as well. I hope that I would have time to talk about some of that in this talk. Now, with the data, I would like you guys to pay attention to the fact that right now in the world we have a lot of data, and the majority of that data, unfortunately, is in the form of a data that we never use or we rarely use. It is estimated that around 80 to 90% of the data that we are producing right now in the world is in form of dark or cold data. And the dark and cold data are the data that we just produce, and most of the time we never use. Hot data is the one that we use most of the time. For example, the communication that I have right now with you in this talk is right now is hot data. You can communicate with me. I will send information. You will send information. Whereas after the talk is done, it will be stored on a cloud, and after a time, let's say in 10 years from now, probably this would be only archived, and there will be less of usage for this video that is recorded. So that's the part that we can call the cold data. Now, the dark data is that data that we probably never use. That includes the images that we take, usually thousands, hundreds of them, and we never go back and even review them, right? Right, so we just store them. Now, the idea is that how do we deal with that? Let me also give you another side of this problem. Now, with the amount of data that we're producing right now, we predicted that by 2040, we might not be able to store all of that data that we're producing on silicon memory. It's just because the rate of production of data is going up exponentially, whereas the refinement of silicon is kind of steadily going up, but it's not going to be able to provide the amount of storage that we need. And this is all about digital data, right? But at the same time, if we look at the cells do have a hard drive, and that's nothing but the DNA that we have in chromosomes or in the genome of the cells. And the principle is fairly simple. The data is on DNA, and then you would have machinery of cell that works with that data and processes the data. So majority of the activities the cell does is basically just processing of that data that is already existing on the DNA hard drive of the cell, right? So the community was thinking about how about we just use these two features that are very similar from the DNA, and how about using DNA as a memory material so that now we store information, the digital information that we have on DNA. And so the principle is fairly simple. You do have the data. You start at encoding. That's your step one. After you encode the data, you only need a map that maps the zero ones or any type of coding algo that you have could convert that to DNA basis. And I'm not going to go into details of that. If in case there is a question, we can touch base on that. But then you would just go and synthesize the DNA. You can store that DNA for a long time. And then whenever you need the data back, you can do sequencing. These are technologies that are enable us that have enabled us to think about these activities. Now, after sequencing, you just only need to recover the data by using a map that is reversed to the first map, right? So it's fairly easy. You just store information that way. Now, for more information about this, you can go and read the paper that we published in Nature Materials a few years ago. This was one of the earlier attempts before the semi-synth bio roadmap came out. And we, over there in this paper, we argue that the DNA molecule can provide better energetics, better information capacity, and at the same time, has a long retention time as well, meaning that the data can stay intact for a long time. Given, for example, we can dig down and take fossils out of soil and take their DNA out and sequence that species that lived years ago, right? And recently I did some calculation and I found that a lot of environmental footprint that we have from semiconductors comes from the refinement of silicon itself. And at the same time, storage of that data also requires a lot of energy, and that has environmental footprint. It is estimated that around 2 billion the amount of CO2 that is being produced is about 20% of the CO2 that the whole process takes just to store the dark data, right? And that's about 2 million trips between just to put it in a concept, that's about 2 million trips between Paris and New York round trip. So that's a lot of environmental footprint or environmental consequence that we're talking about. And DNA just does that essentially for free because cells are mostly dealing with DNA in a very green way. It's actually a biological material as we speak. So anyhow, so those are the motivations for us to think that DNA could be a good material as an archival data storage. Now, right now, the way that we do deal with DNA as a archival data storage is by just translating or mapping the data towards DNA bases and then go and synthesize that. But the synthesis right now is very much chemistry based. It's very slow. It's very costly. But more importantly, it produces a lot of toxic waste. So and it's also not rewritable, editable, or reversible. And there is also security issues. This is the three red points that I have here are the focuses of my lab right now. So the first thing that we came up, just to put it in concept, that environmental consequence is huge. If we are to put data on DNA, let's say by 2025, we put all of that data on DNA, we're going to produce so much waste that is going to destroy the earth. So we got to look for a alternative solution. And one of the solutions that our lab came up with was to just use it already existing DNA. We just define domains on that DNA. It's very similar to a punch card concept. And that DNA strand has let's say domains that we can consider intact or state zero, no data, or zero, data zero. And then the only thing that we now need to do is just to go on those specific domains. If we have the control over the domain, just go and mutate that domain. And then the data is been written in a form of mutation. So we use CRISPR-Cas9, dCas9 to block and activate the specific locations on that DNA domains, and we use APOBEC to convert Cs to Ts. This way, we made mutations intentionally, similar to the mutations that happen in nature, but now we intentionally mutate specific bases, and as a result, we would be able to write information. And long story short, we have done that. This is a paper that we published in 2023, and we stored the title of that paper on DNA just using this DNA strands and just trying to mutate the bases. So I'm not going to go into detail of this, but there were errors all over that data, and we needed error correction. At the end of the day, what I hope you can see is that on the right side of the graph, on the lower graph, you would see that we were able to recover the data with the green dots in the true state. That means the file was recovered completely. So that's one way of storing information. Now we're trying to reduce the size, increase the capacity, and make it faster and all of that. So this is a chart of our microfluidic system. So hopefully this would be something that looks like a flash memory, probably a bit bigger than that, but hopefully would be a molecule, a device that we can store information. So with that, I would like to switch gear a bit, and that would be where probably this would be a bit more connected to the previous discussion that Joe had. And this is where we try to use DNA molecules as logic operators. And so simply, if you have two DNA molecules that they have interactions with it, or they have the potential to have interactions with each other, in another word, they're complementary to each other, you would expect that they can, in a spontaneous reaction, just get together and make a third molecule, as I have it on the left side of this slide showing. That's nothing but a similar activity that a logic gate, and in this case, an AND gate, would do. You just have two inputs and it produces an output. Now we can use this for many different applications, and I have listed some of that here, but we mostly are interested in the diagnostic and also switching activity, and I'll show you why that is important. Now, let's start with this. This is a map of a metabolic pathway of E. coli. It's a very simplified version, I would say, but it's still very, very complicated, right? So reading this is pretty difficult, and doing things with this system is pretty difficult. This is the language of cell, right? So looking at this map, you would say, well, how would I be able to manipulate a cell, right? But now I'm going to show you something that makes everything much more simplified. And that is, just look at one of those nodes. Let's say we look at only a lac operon node, and let's see how that performs. This is the language of cell. If I want to do anything with the cell, if I want to manipulate the cell, I need to know the language of the cell and speak it, right? So let's see what the language is. First, the language, knowing that the lac operon is nothing but a couple of gates, right? So you have a NOT gate when you don't have glucose, an AND gate when you don't have glucose and you have lactose in the environment. The preference of the bacteria is to use the lactose as a food, and it wants to survive, so it produces the beta-galactosase enzyme that cuts the lactose, right? But that's not very energetically, and it's not very friendly for the cell. So it prefers not to do that unless glucose doesn't exist. So it's only a survival mode that the bacteria would use the lactose as a food. But it's all based on the logic that we have here. It's a logic gate operation between different parts of those circuits. And so that's the language of cell. So we now know the logic is the language of cell. We only need to speak it. And as it was shown by Joff. They have way more complicated system, but we also try to speak that, to do that we came up with making structures, and these are nanostructures made out of DNA. It's fully made out of DNA. There is nothing else. There's a couple of fluorophores on the structures, but that's only for our studies. However, so these molecules, they are completely made out of DNA, and they have, so this is specifically a box-shaped structure that has a hollow interior. And the idea is that we're hiding something in that interior that would interact with biology. And at the same time, this box has a lid that can be open, and the opening of the lid is when we introduce the keys. And the keys in this case is in this publication that we had in ACS Nano is a couple of breast cancer microRNAs. The concept is just fairly simple. You have an AND gate, the two microRNAs existing, that the cage will be open, and as a result, the material inside the cage will be accessible. And this is a fairly small molecule. We're talking about something that is the size of a ribosome. So you can probably be doing some manipulation of cell inside the cell, if it's uptaken. Now, I have an animation that shows how the system's supposed to work. This is a cage structure that we were talking about, a bit bigger for presentation. But then if you have the microRNA of the keys, so initially when the molecule, that yellow molecule, is supposed that's a biological molecule, wants to interact with the material inside the structure, it would not be able to do that unless I have the keys, which in this case, again, are microRNAs of cell. And these microRNAs are specific for cells. Say, for example, it is very specific to breast cancer. Now, it will open up the structure, and as a result, that molecule has access to the interior of the structure. And now in this case, this is a restriction enzyme that cuts the restriction side. The green side of that animation, that drawing is the restriction side. So nonetheless, what we expect is that the enzyme would have access to the interior of the cell of the structure if the microRNA exists in this environment. And that release of molecule could be doing something for us in the cell. That could be, for example, expression of a gene, or it could be a drug that we just hide inside these structures, and it will be released. So that's one step towards intelligent medicine and to some extent personalized medicine. Now we of course showed that this is possible. We made a structure that is very similar to a chromosome in terms that it will wrap around a very essential regulatory element of a gene. And if you don't have the keys, that in case again are microRNAs, you would not be able to see expression of that gene. And in this case, RNA polymers cannot do any activity. But when we do have the keys, it will produce the RNA. And two of my brilliant students are working on this project right now. But the idea is at the end is that you would have the RNA being produced if the keys are existing. And say I have a cancer cell that has specific microRNAs. My cage goes into those cells, and in case that the microRNAs exist in the cancer cell, it will open up, and as a result, you would have the expression of genes. Say, for example, the cell doesn't have P53 pathway active. We can use the gene P53, it's an external coming, and then the expression of that would be resulting in the apoptosis of the cell. So the normal cells do not have those microRNAs, hopefully, and as a result, they would not result in expression of that gene. So that's the goal, the final goal that we have. But initially, we have done some experiments that shows we can control that gene expression to a good extent. We can limit the expression of the gene if the microRNA mimics don't exist, and if they do exist, we do have expression of gene. And it also is a reaction that is concentration dependent. In this graphs that I have here, that shows our data supporting this idea. Now, with that said, there are a lot of things that we need to do from this step. We need to make sure that these structures can be uptaken by the cells. We need to make sure that we can also control these activities inside the cells. Um, and a lot of different types of studies that we need to do. We want to make sure that these structures are not they're not activating immune response from the patients. With that, I want to go to this slide first and more foremost. As I mentioned, there are a lot of different studies that we still need to do, and I'm constantly looking for students, whether undergrad, master, or PhD students and postdocs. Our lab are welcoming everyone. So, in case you guys are interested, I would welcome communications. Please either email me, that's my preferred way, or go to the website and learn more about us. But the best way to connect with me is just through my email. Fairly easy. My first name, the first letter of my first name, and my last name at ncat.edu. With that, I would also like to thank all the agencies that have supported these works so far. Yeah, taking questions in case that there is any.
Fantastic. Thank you so much. Absolutely great talk. Before I go to the audience, Geoff, do you have any comment or questions?
I was really curious as you're building these cages, what kind of size cutoffs have you achieved, and what do you think the limits are where are the limits today, and where do you think you can get to? It's very
Yeah,
interesting with the AND gates built into that.
very, very good question. So that the example that I showed here, this is a bit more complex system, and that's about the size of a ribosome, so 18 nanometer roughly. The smallest that we could achieve was with this newer design. This is just only something that wraps around the promoter, and it very much depends on the size of the promoter that you want to block. But the one that we have been using is pretty small. It's about 9 nanometer. And I would say that is probably the lower limit. Beyond that, probably we would not be able to construct these structures very efficiently. There would be a lot of leakiness coming from them. But so that's our probably lower limit.
And
Now,
so
higher limit.
for the larger one that you showed, what is the small, like if you were to put molecules in it for maybe
Oh,
a slightly bigger one, then what is that constraint?
Yeah, so it very much depends on how much DNA you want to put in and what your template is. But I would say right now, so the common template that people use for DNA origami is about um, uh, uh, 8 kilo bases. So that is around a molecule, or that would give you a cavity that is about a um, I would say a molecule that is at most 20 to 30 nanometer. Now, that said, you can make a system, specifically framework DNA structures that are way larger. So you can put molecules up to a few hundred nanometers. The upscale would be probably with the normal, like you don't really do any modification, any tough work, would be around, I would say, probably 400 nanometer, 400, 500 nanometer. Now, that said, you might be able to, you can, there are some examples in the community that they have used a very larger template, and those are lambda DNA, for example, and that increases the size drastically. You can imagine the size that I'm referring to can be multiplied by 10, a factor of 10. So probably about a micro to 5 micrometer molecule could or construct could be put in. Now, controlling that is a big issue in my opinion. So there is a lot of challenge, but theoretically, I think that the upper and lower bound is between probably 10 nanometer lower, and the upper would be around a micron. Um,
But leakiness. So let's say you take the prior AND gate.
Yeah.
can you like you're talking about a macromolecule. As you shrink the cargo,
Yeah.
oh, what's the smallest cargo that you could package that won't leak out?
It very much depends on the design. Um, for this structure that I have it on slide 19, that is a piece of DNA, strand of DNA. And for this one, I would say it's just that strand of DNA, right? So it's about 2 nanometers, the width of it, and the length of it would be something that I can that I can change it right easily. Now for this structure that I'm showing on slide 17 specifically, my calculation, the way that I looked at it, and this is a previous work, but the way that I looked at it in calculations and also the experiments that we did, a molecule of about 7 to 10 nanometer would not leak out.
Okay. No, no, no. That's
But
that's exactly
yeah,
what I was curious
yeah,
about.
yeah.
That's really
But yeah. So, but smaller than that, there would be probably
sure.
leak some leak that happening. Yeah.
Super
But
cool.
it's again, this very much depends on the design because if I'm opening up the sides, right? Because I want to have a bigger real estate, the gaps are going to be bigger. So, but we can control that. I would say we can pretty much in the community we have shown that we can control that pretty well. Now, not 100% of course, right? It's not going to work that way, but yeah, to some extent. Very good question. Thank you.
Geoff, do you have any other comment or question? Okay, so let me ask one question. I found this is very interesting, also coincidence. The first speaker of this series, 3.5 years ago, also talking about Albert Kuhn,
Yeah,
also, talking about, DNA storage, happened to be also, I believe, North Carolina. Uh,
yeah,
and the question is, you mentioned in the middle of your talk about a lot of challenges, including interestingly, potential problem, environmental problem as well. And over the many years, I listened to many talk about DNA storage, and then whenever I listen to those kind of talk, like sometimes imagine in the future. I mean, we potentially have DNA storage, basically you know uh kind of realized. And then, but you also mentioned, some, challenges, but do you have any kind of vision, uh, as a community, how we gonna solve that problem, more drastically faster than we are currently doing? And then what would be your insight about those kind of things? Because I'm curious about, always, I
yeah.
mean.
Again, very good question. And I think that is a fundamental, uh, like existence, the fundamental question for for existence of this field moving forward.
Mhm.
Um, and we're constantly, I think, looking at that. So I'm hoping that this slide number 9, um, is listing. I'm hoping that this is slide that lists some of the challenges would be a good map forward. Now again, I'm a very small fish, so for what it's worth, what I think the community needs to do to make the DNA memory as a alternative material for data storage happen, what makes it um, feasible and industrially attractive would be if we have, so right now we have a huge latency, which means we're the system is very slow, whether you're writing or reading, both are
Mhm.
very slow.
Right.
Um, the second is that it takes a lot of computation just to do small jobs right now. So that's a lot of energy that we're spending. Um, and and so right now, a majority of the activity was focused on just synthesizing pools of DNA. and write the information on that and then sequence that, which is the most convenient, the easiest way to do it. However, the issue with that, so the first two is just that we would need to work on better synthesis, faster synthesis, cheaper synthesis, and probably the same for sequencing as well. Now, I would argue that since I'm proposing DNA memory would be mostly appropriate for archival memory, because the speed probably would never be able to match the silicon for many reasons, and including that the fact that it is a chemical biological molecule. But I think as an archival material, which is a very good argument still, because majority of data is archival,
Mm-hmm.
right? We can release the capacity for silicon by just using this as an archival. So a hybrid system basically would be a good, I would say, solution. Now, anyhow, so we need a better, a faster system for synthesis, faster synthesis, a cheaper synthesis. Um, and so that's for the DNA. But at the same time, if we go and write on DNA and de novo synthesize DNA each time that we have a new data, that is not scalable. Like the example that I have is, say I have a hard drive on my device, my computer, right? And everyone has that. Say this hard drive comes with information already pre-punched on it, right? Anytime that you want to add or edit an information, you gotta go and buy a new hard drive from that main company, right? While that company probably would love that, however, the consumer would not like it because you would need to go to the source. It would be taking time. It's also expensive, and you have to, each time you are editing only one bit, you got to go to the source and buy. So we need to have a system that gives the scalability that comes from, in my opinion, comes from the mass production of a raw material that is empty in per se, like a hard drive that you buy and it has no information on it or very minimal information on it. And then you would write your own information on top of that. That's why we were motivated by the system that we proposed, that you have a strand of DNA that is empty. And now you would use biological molecules such as the CRISPR-Cas9 ApoBix system to just modify that DNA. And as a result, you would in mass produce the template, empty template, and then write information on it. That still is very slow though. So that's the issue that we have, right? So we gotta work on that. I hope that answers the question.
Yeah, that answered question, and also that remind me of my conversation, very interesting conversation yesterday when I visit UC San Diego. I talked to Terry Hua. He is the very renowned world class, originally doing physics, but now bio stuff. And then the one interesting comment he gave me was, we doing synthetic biology now, I would say, almost, 24 years or 25 years, roughly since, 2000. And then he asked uh, whether we are really doing predictive, quantitative biology. And then he said, uh, we still kind of it's just baby step. We just putting into the field because we are not necessarily have any. I mean, we have some tools, but not necessarily we are predicting anything uh, accurately with the real number in it. So
Yeah.
that's a very interesting, insight I, got. And then we're talking about a bunch of different things, including physics, chemistry and other field, and the relation to the biology, etc. And then he mentioned he also worked for the company that's an electronic company, and then we are comparing our biological system with the electric or electronic circuit. And then he just mentioned whether that is really true or not, and that's another thing. kind of. I agree with him in a sense because we're talking about multiple different things, but we have lots of challenges, but that means also we have lots of opportunities in the future, and I hope young people would solve many of the problems we currently having. In that way, we move forward much quicker than we wanted to see. So that's why I'm asking that question. We are now at 11 Central Time, and then let me check whether we have question from audience. I don't see anything now. So let us close, and then we probably have informal chat afterward. Thank you again very much for your wonderful talk. It's fantastic. And also thank you all for joining us and staying today. We'll meet again on January 23rd, Thursday, the same time, the same Zoom link. We'll have the Professor Tilman Weber at Technical University of Denmark, and also Anna Faber at the University of Western Australia. I believe now this is really global. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks again, and I stop recording. Just 1 second.